Good morning to our friends in Brazil. Good afternoon to everyone watching from the UK. It is with great pleasure that the Embassy of Brazil in London launches today the Science in Brazil project. We have conceived this project with two objectives in mind. The first is to celebrate Brazilian researchers' contribution to science. The second is to attract international attention, international researchers to Brazil. To achieve these goals, the Embassy will host a series of webinars that will showcase cutting-edge research being developed in Brazil. The first of them, Science in Brazil Saves Lives, which will start in a few minutes, will focus on health and medicine. After everything the world has been living through since last year with the pandemic, it seems almost natural, most obvious, we should start with the Brazilian contributions to science in this field. We cannot talk about innovative science in Brazil without talking to scientists, to the scientists behind it. Therefore, I would like to thank Dr. Esther Sabino, Dr. Nuno Faria, and Dr. Fernando Sendes, leading researchers in their fields, for taking the time to speak with the audience about your research. In particular, I would like to thank Dr. Carlos Henrique Brito Cruz, not only for moderating today's seminar, but mainly for his participation in conceiving and organizing this project. The idea behind Science in Brazil came from a conversation we had in which Dr. Brito mentioned how little the world knows about innovative research being developed in Brazil. So Dr. Brito, the Science in Brazil project would not exist without your valuable insight, assistance, and suggestions. Other partners have also proven instrumental for the project. The São Paulo Research Foundation, FAPESP, is one of them, and we are very grateful for their support. So I'll pass the floor then to Dr. Brito to introduce our first speaker today and start the seminar. Enjoy.
Thank you very much, Ambassador Arruda. It's a great pleasure to be here at this virtual symposium about science in Brazil, which is the first instance of a series. And today we are going to be covering topics related to health science. And we have three speakers. They will make brief presentations, 15 minutes each. And then we are going to have a Q&A session. The speakers for today are Professor Esther Cerdeira Sabino, who is a professor at the University of São Paulo, and Professor Nuno Rodrigues Faria, who is a reader in virus evolution at Imperial College and an associate professor at the University of Oxford. They will both talk about SARS-CoV, coronavirus, virus sequencing, and topics related. And then the third speaker is going to be Professor Fernando Sendes, who is a professor at the University of Campinas, also in the state of São Paulo in Brazil. He is active in neuroscience and neurotechnology. So we are now going to start with the presentation by Professor Esther Cerdeira Sabino, who is a medical doctor, doctor in science, and besides being a professor at the Department of Infectious Diseases at the University of São Paulo, Professor Sabino leads the research center, which is the Brazil-UK Center for Arbovirus Discovery, Diagnosis, Genomics, and Epidemiology, CADDE, which is a center that was created through a collaboration between the São Paulo Research Foundation and the Medical Research Council in the UK. I believe it was started 4 or 5 years ago, and the arbovirus in the title of the center comes from the Zika epidemic and dengue. And then with the pandemic, they veered their direction towards the SARS-CoV virus. So, Professor Sabino, please, you have in the old times, we would say you have the floor, now you have the screen.
Thank you for the invitation. It's a pleasure to be here and show how important is this collaboration, especially this international collaboration for the response of any epidemic. And I'll start showing a little bit my story and link how Richard collaborated with Nuno Faria. And I was training in San Francisco in 91, and during the AIDS epidemic. And during that time, I could see how epidemic can completely change the way you do something. And in this case, how transfusion medicine changed completely, because the virus entered US in 1977, but was only recognized in 81. And the virus was only discovered 2 years later, and a test to use in the blood bank was only available in 84. So during this period, there are lots of transfusion-transmitted cases of HIV. And this made the NIH respond with a huge program that's called REDS, Retrovirus Epidemiological Donor Study, and they didn't want it to happen again in the US, because almost 1% of the units, like in San Francisco, were contaminated. So I was trained during this period, and in 2006, they opened, I was already back in Brazil, they opened a call for international sites, and we could submit one and win. And this is a long study now with almost, the American study is 30 years, and we are gonna be almost 20 years when we have already went in 2025. So this is when I was there. And so during this period, what we could establish is a nice network of blood banks, and we could learn how to extract the data and run studies in parallel. And we have centers from 5 different regions of Brazil. And one of the things that was important, and the first time we worked with this group in a response for an epidemic, was the dengue virus. This is because dengue is already, they are starting to see dengue in Florida and Puerto Rico. So this is a problem for the US, and they wanted to know whether dengue would be transmitted by blood. So this was a common interest, and we did this huge study when the dengue-4 entered Brazil in 2012. So we chose the cities with higher chance of having the epidemic that were Recife and Rio de Janeiro. And with this study, we had to include 4,000 blood donors, informed consent, and we did around 30,000 NAT testing to arrive to 120 dengue cases to learn how this, what was the frequency of transmission by dengue, which is around 40%. We didn't really detect any impact in the recipients. But this also showed, after the study, that was the main thing to understand how transmission occurred. We see that this data could help us understand how the epidemic was in the city of the Rio de Janeiro, that we could follow the complete epidemic. And also, we got another grant to sequence those 120 dengue, and that's how we link now with the group from Oxford to analyze those sequences and understand how it spread during that period. So after that, we've been working also to understand these epidemics in the blood banks. So for example, we are now testing systematically around 400 units in pools from each one of the sites for the 3 viruses. So we can see how long the Zika stay in Rio de Janeiro. These are the reds here in the top. Or now we can see the epidemic in dengue in 2019, and it almost reached 1% of the units in certain weeks. So it gives us an idea and also how we can use this established infrastructure to understand new epidemics. So because of that, when the SARS-CoV-2 started in Brazil, we thought we could really use this infrastructure to understand the epidemic. And the epidemic in Brazil for SARS-CoV-2 was somehow we lived a continuous epidemic. It started as like a plateau. The first full year we had cases, decreased a little bit in the end to start back again in the gamma epidemic. But it didn't flow the same way in each one of the cities. So some cities like Manaus and Fortaleza have had huge peaks, while some regions like São Paulo or Curitiba were more like a plateau with these peaks during this year. So the idea we had, well, let's use the blood centers to understand how many people got infected. And to do that, we use tests. You can detect antibody and measure who really got infected, because the antibody will appear in people later, even if they don't have symptoms, some of them might show up. And so you can measure better how many people were exposed to the virus. So we could do it with 1,000, we thought of testing 1,000 blood donors per month in each site. And one thing that's important in Brazil, we could go back, because in Brazil it's mandatory for the samples to be saved for 6 months. So even though the tests were only available in June, July, we back and understand completely how the epidemic moved every month. So, and we also try to have a good geographic coverage from the cities. So we developed a system to overcome the problem that the donors near the centers donate more frequently. So we choose and we organize, so we had a better view of the city, the way the sampling was done in these studies. And we did it for all the regions. So that the same, the tests arrived early in July, and São Paulo and Manaus are more ready to go back and test everything very rapidly. So the first paper, we had more the data from only these 2 cities, although we were testing the others. So we soon realized that Manaus had a really big epidemic. The antibody levels were very high, but we saw already that the tests we choose really declined, antibodies declined very rapidly. So to understand the attack rate or how many people got infected, we had to do a correction. And by doing so, we reached the point that about 70% of the people by October had been infected by SARS-CoV-2 in Manaus. And this caught a lot of attention, this paper, because this level is higher than what is the theoretical level of herd immunity. So for viruses that have a R naught, or how many people got infected by one infection, that's around 2 to 4 for SARS-CoV-2, this level when the epidemic started to decline is 66%, around that. So it doesn't mean that the epidemic ends when you reach the herd immunity. The epidemic goes until the final attack rate, normally for a virus of with this R naught, would reach 95%. But probably if the immune response were durable, probably we should not see another epidemic this size. So in this sense, Manaus was a sentinel to understand what happens after you reach this proportion of infection. That's why there was so much interest about this paper. But as soon as the paper went out, that was in December, we had starting another big epidemic. What put us a lot of pressure is to see whether we have made all the corrections and whether we are sure about the size of the first epidemic in Manaus, because this had a lot of consequences. But indeed, and probably Nuno will show more, the increase was associated with a new strain, a new variant that's initially called P1 and now gamma, that had several mutations and probably a much higher capacity of transmission. But we were quite worried to understand whether we had made all the corrections or all the assumptions in our models correct. So we went back to try to understand exactly how to correct this. And I think this is really important to understand rates of reinfection and what we were really measuring. Because what sometimes we don't pay attention is that the response may be very different according to how the patient had their disease. And what we are working in the blood bank are with mild and asymptomatic cases and not severe cases. And this, we went back to see a lot of, and tested several repeat donors from Manaus, so we could understand how was the decline of the antibody among the repeat donors. And this first showed that indeed, the distribution of antibodies is different than the cases, the people who have PCR positive and symptoms, they have lower response, and that response declined much faster than the plasma donors. So now that we have this in hands, we could go back and test all the cities, and now we know the attack rate, and understand that for some cities like Belo Horizonte and Curitiba, the attack rate was similar to other regions in Europe, but some other regions had a much higher attack rate, including Rio, São Paulo, that reached around 50% or sometimes more by the end of the first wave. And also with this kind of data, we can figure out what was the infection fatality rate, how many people die per infection. And these are shown here first that indeed, the higher is the prevalence in the blood donors, the higher is the deaths when you control for the age, because the cities have different age structures. And here just show without correcting it. So cities that have more older people had a higher IFR, but when you correct by age, you particularly see similar IFR in the different, according to the age of the standardized age. So that was an important result to show what was the infection. The other thing is that is about the vaccination. So the vaccination started a little bit later in Brazil. In the beginning, only the people older than 6 years old were got the vaccine, but by May we start having the vaccine given for everybody, and the population really responded. And today Brazil has more people vaccinated, completely vaccinated than US, and even with the United Kingdom, we have at least the first dose given in more people than in the UK. So the blood bank, the Delta started in Brazil in September. The majority of the cases were Delta by September, so I started earlier, but in September was already 100% of the, but we are not seeing a third wave by the Delta. And maybe, we don't know, we are trying to understand that the antibodies as seen by the blood centers might give some clues of what's going on. So here, you see what was the level of the antibody now with a new test, the NTS, that doesn't decline so much. Before in November and after the epidemic, the gamma, you see that increase the proportion of the positivity, but you don't see a lot of people with high level of antibodies. But when we start vaccinating, the level of antibody becomes very high. This is Manaus, and we are seeing this for all the other cities. So maybe we might have a better evolution of the Delta. In Brazil, we are hoping that this might happen. Okay, so in conclusion, I think I just want to show how that we can use blood banks, and this is an important source of information during an epidemic, and how important is international and long-term scientific collaboration for a rapid response during an epidemic. So these are the groups, the support we got, these two key studies, CADI, that Nuno will explain more, and also the REDS program. And thank you very much.
Thank you very much, Esther. That was a very informative and really, really interesting talk, not only showing the transition between different viruses ending up at this moment in the SARS-CoV virus, but also highlighting the relevance of the existence of a network of blood centers, which you found a way to use as kind of sentinels for the infection across Brazil. Very, very, very interesting. So I'll pass to our next speaker, and we are going to have the Q&A and discussion in the end. So the next speaker is Professor Nuno Rodrigues Faria, Master in Science, Doctor in Philosophy. He's a reader in virus evolution at Imperial College, London, and an associate professor at Oxford University. And Professor Nuno coordinates the UK side of the Brazil-UK Center for Arbovirus Discovery, Diagnosis, Genomics, and Epidemiology, the CADDE, which is directed in Brazil by Professor Ster. So, Nuno, the floor is yours.
Can you see my?
Yes, it is here.
Unmute. Okay. All right. Thank you very much for the introduction. Hello, everyone. So, bom dia. It really is sort of an honor to be here today and to present some of the work we have been doing over the last few years in viral epidemics in Brazil. I have included here this painting from Tarsila do Amaral, as the close contact between animals and humans shown here is a prime introduction and also requirement for the transmission of zoonotic pathogens. But what are zoonotic pathogens or zoonoses? So zoonoses is essentially any disease or infection that is naturally transmissible from vertebrate animals to humans. And we know that there's over 200 of them, and most are caused by viruses. So these comprise a large percentage of new and existing disease in humans. And this include, for example, rabies, Zika virus, yellow fever, and SARS-CoV-2. Most of these zoonoses can go undetected for long periods of times, but others, like Zika and SARS-CoV-2, can also lead to major pandemics, which are typically monitored using genomic sequencing. Due to limitations in surveillance and lack of funding, there are many estimated missing zoonoses across the globe, let's say. And since traveling became so much faster in the last decades, we are living in an epoch where new zoonotic viruses can very rapidly go global. So let me start by taking you back in time to about 6.5 years ago. So in March 2015, a mysterious cluster of infections was identified in the northeast region of Brazil. 10 months later, in January 2016, the first Zika virus genomes were shared with the scientific community, including one genome from a newborn with microcephaly. So to track Zika and to better understand its association with microcephaly, we set out the ZEBRA project, Zika in Brazil Real-Time Analysis, a collaboration between USP, IMT, USP, Fiocruz, Instituto Ivan Chagas, and several UK institutions as well. So during a 2-week period, we traveled 2,000 kilometers on a mobile lab and tested over 1,200 samples from public health laboratories across 6 states in Northeast Brazil. And we generated 50 Zika virus genomes on the road. Genomic data, confirmed case counts, and results from additional mosquito surveys were shared in real time with the international community. So here in the center, on the left side, is a phylogenetic tree of Zika virus genomes, the equivalent essentially to the time-scale genealogy of the virus. The tips here of the tree correspond to virus sequences, and the colors of each branch throughout the phylogeny correspond to the virus ancestors present in different geographic locations. We found that Zika was introduced in Brazil from French Polynesia by a single introduction event that we could date back to January 2014. So importantly, this meant that Zika circulated undetected for around 18 months in Brazil. The virus spread rapidly in tropical and subtropical areas where the mosquito vector Aedes aegypti was prevalent and became well established in 87 countries across the world. Yellow fever is also a mosquito-borne virus that is endemic in Brazil. So before the 40s, it used to be 1940s, it used to be transmitted also by the Aedes species mosquitoes. But after that, the virus shifted his behavior, and it seems now to circulate predominantly in forested areas, being transmitted by mosquitoes that live in the tree canopies and feed on non-human primate blood, essentially, in what we call a sylvatic transmission cycle. But in 2017, the number of human deaths was the highest on record since 1920s. So we responded to a request from Pan American Health Organization to investigate whether the ongoing yellow fever outbreak was shifting towards a more urban transmission cycle. So within 6 days, data again was generated, analyzed, and interpreted. We found that all yellow fever human cases were ultimately the result of independent zoonotic events between non-human primates and non-vaccinated humans, with this being mediated by sylvatic mosquitoes. By the end of the outbreak, yellow fever had spread to São Paulo and Rio de Janeiro municipalities, where most populations were unvaccinated, claiming over 2,000 lives. In this process of investigating major outbreaks in the past, our teams trained hundreds of researchers across Brazil and beyond. So our projects involve training at Fiocruz Bahia and Fiocruz Rio and Fiocruz Amazonia, but also FUNED and the UFMG in Minas Gerais, and at IMT USP at São Paulo, including also several institutions overseas and the Ministry of Health in Angola. And by April 2019, actually, we initiated our CADE project. So CADE, in Portuguese, CADE reads as CADE, essentially means where is it, what is it, in a reference to the work we do in relation to virus discovery and virus origins. A big part of our regional team met during the 2015-2016 Zika epidemic, and CADE currently operates as a collaborative network of labs across Brazil, with an epicenter at IMT USP, led by Esther, and also at UK institutions with an epicenter at Oxford University, led by myself. So in gray, I'm showing some of the institutions that collaborated with us, that started collaborating with us after the beginning of this award. So we maintain a strong focus on training and capacity strengthening, and we welcome everyone interested in partnerships and collaborations. And the focus really of the project is to integrate genomic, epidemiological, and environmental data in real time to help guiding public health interventions. We are focused on pathogen genomics, mathematical modeling, clinical studies, public health, and operational research. And we were originally prepared to investigate in detail mosquito-borne viruses in 2019, but given our relatively small scale, which comprises a team of over 30 core people, we were rapidly able to shift our focus and our scope in February 2020. What happened in February 2020? Well, this figure presents a timeline of the SARS-CoV-2 pandemic in Brazil. The boxes here sort of highlight some of our key SARS-CoV-2 studies and some of our key data sharing initiatives as well. So given that we are into the 22nd month into this pandemic, nearly 2 years since the first reported case in Brazil in February, we now have a good idea of the number of cases in Brazil and the diversity of circulating lineages. So the national epidemic curve, which is shown here in blue, shows essentially 3 phases with their increases and declines. The first phase started really with the detection, sequencing, and analysis of the first SARS-CoV-2 genomes cases in São Paulo. It was also the first case in Latin America, which we called SPPR1, that was identified by our team on the 28th of February 2020. So the second phase started with the detection of the P1 or gamma lineage, which CADE detected in Manaus on the 10th of January 2021. And we have recently entered, like Esther mentioned, the third wave, the third phase, in the period of continuous decline in the number of cases coinciding with the Delta variant of concern, and also, most importantly, perhaps, the increase in vaccination rates. And hopefully this decline will continue and will be maintained throughout the next months. A few weeks before the first detection of SARS-CoV-2 in Brazil, we used flight data to identify the routes of SARS-CoV-2 introductions in Brazil. This is essential for pandemic preparedness. So given that all population in Brazil were a priori, and in the world were a priori susceptible to this virus, the frequency of introduction should be proportional to the volume and the location traveling. So in early February 2020, we found that nearly half of all initial cases would be arriving at São Paulo's largest international airport in Brazil's largest international airport in São Paulo, and this case would be coming from Italy. In contrast, for example, very few introductions, less than 0.1%, would be expected in the north, in Manaus, the largest city in the Amazon region. So I think it's worth highlighting here circumstances around this detection of these first SARS-CoV-2 cases. So on the 26th of February, we received the first SARS-CoV-2 suspected case in São Paulo for laboratory confirmation and sequencing. And it took the team 48 hours to go from sample to PCR detection to genome sequencing to analysis and interpretation. I think this really was unprecedented, particularly when we consider the delays in detection during, for example, the Zika pandemic. The sequencing was done by Esther, Sabina, and Jacqueline and their team. And here is a cartoon by Mauricio Souza highlighting this initiative. And Jacqueline, a postdoc in our team, was later immortalized by Barbie Mantel as well. But it is really important to consider that this was only possible due to experience and capacity that was built since 2016. And as a follow-up of this initial case investigation, we joined forces with LNCC, with Unicamp and UFMG, and we generated the first large genomic dataset of SARS-CoV-2, publishing a seminal paper with 148 co-authors, 80% of them from Brazil in Science. I think it's also important to remember why exactly do we need genomic data during pathogen outbreaks? Why is it so important, and what does it allow us to address? So CADA members also contributed to the WHO sequencing guidelines for SARS-CoV-2, which I very much recommend for all labs in the call conducting genome sequencing activities around the world. But in brief, genome sequencing is critical to identify which pathogen is causing an outbreak. We can analyze early data to understand origins and zoonotic events related with, for example, repeated cross-species transmission, as we've seen for yellow fever. Sequencing is also critical to design diagnostics and vaccines and to track mutations of interest that might affect their effectiveness. And overall, it complements epidemiological assessments of SARS-CoV-2 transmission. Analysis of sequence data can tell us, for example, where introductions are coming from, how the virus is spreading within a given location, and how to target interventions at different scales. And that scale could be within a country, within a city, or even inside a hospital. So here is an example of how we use genetic analysis of sequence data to uncover the early patterns of SARS-CoV-2 diversity in Brazil. First, we found over 100 independent introductions, each represented by these red circles in the phylogenetic tree, with some eventually leading to larger clusters of transmission corresponding to transmission in the community. Particularly, we found 3 lineages that comprise 76% of all genetic diversity circulating in Brazil. One of these lineages was then the precursor of the Gamma variant of concern that I'll be talking in a few minutes. But second, we estimated that it only took about 2 days to detect the first case in Brazil. This is encouraging. So as I mentioned earlier, it took us over 18 months to detect the first Zika case in Brazil. So such a rapid detection of the first SARS-CoV-2 lineages in the country represents and reflects really the importance of improved preparedness of the most at-risk locations to detect early cases. And finally, sort of in the upper panel, what we have here shows the density of international virus migrations to Brazil in blue versus the internal virus migrations within Brazilian locations in red. It's really interesting that just from genetic data alone, we could detect an early initial period of international reports, followed by a subsequent period of virus lineage movement within the country. So by late December 2020, we started noticing a rapid increase in hospitalizations and deaths in Manaus after a large estimated attack rate, as mentioned earlier in Esther's talk. By then, there were only 6 genomes available from Manaus. 5 of them were generated by our team. So we rapidly put together a task force to investigate virus diversity during the second wave there. We were expecting to find the alpha variant of concern that was detected just a few weeks before that in the UK. But when we sequenced about 200 cases and found a local cluster of SARS-CoV-2 sequences with 17 unique amino acid mutations that we named as P1, and that was later denominated as the gamma variant of concern. We found that 10 of these mutations were in the region of the spike protein that the virus uses to bind receptors in human respiratory cells. And we found some evidence of increased transmissibility. So this virus lineage was spreading faster. The earliest retrospective identification of this new VOC, or variant of concern, preceded the large increase, which is shown here in an arrow, preceded the large increase, the number of SARS-CoV-2 deaths in the second wave, and replaced literally all the other non-P1 lineages. Importantly to say, these results were found in the evening of the 10th of January, communicated on the 11th of January to local teams in Manaus, also to Pan American Health Organization and WHO, and the report and data were published online on the 12th of January in Virological. And I think also important to say that the continued monitoring of gamma following this study showed exponential spread, took place in many other locations outside of Manaus, and within less than 3 months, the virus was circulating in all regions in Brazil. It has now been detected in over 71 countries worldwide. Importantly, we also investigated transmissibility versus immune escape for gamma using a two-category mathematical model that incorporated the waning of immune protection following infection and the estimated timing of the P1 emergence in Manaus from our genetic analysis, which we estimate around mid-November 2020. We then fitted this to the proportion of P1 cases detected through our surveillance efforts and to mortality data from Manaus. But essentially, all our results pointed to altered epidemiological characteristics of P1 in comparison with non-P1 lineages in Manaus. We saw that P1 was spreading 1.7 to 2.4 times faster than local non-P1 lineages, and that P1 could evade 21 to 41% of protective immunity elicited by previous infection with non-P1 lineages. So in other words, in 100 people were infected in Manaus last year, somewhere between 21 and 46% of them are susceptible to reinfection with P1. And we also found an elevated risk of mortality for P1 compared with non-P1, but we cannot really determine yet whether the estimated increase is due to P1 infection itself or to stresses on the Manaus healthcare system, biases in reporting, or a combination of these factors. But more detailed investigations are still ongoing. So fast forward until November 2021, until now, Brazil has sequenced about 0.2% of SARS-CoV-2 confirmed cases, with over 70,000 virus genomes shared in dedicated databases, specifically in GSA. So in the current review we're doing, we looked at SARS-CoV-2 sequencing intensity and turnaround time, which is defined as the time it takes from a sample to sequence to deposition in dedicated databases. And we found that sequencing capacity for SARS-CoV-2 depends critically on the investment in research and development in each of the countries analyzed. And we analyzed data from 167 countries. Importantly, we also found that previous pre-pandemic experience in generating sequencing data, for example, for influenza, was linked with more robust genomic surveillance capacity for SARS-CoV-2. So what these findings suggest to us is that the more we boost genomic surveillance worldwide now, the higher the chances we are in a better position to respond to future pandemic threats like disease X and to control it before it reaches pandemic proportions. So let's do this. In conclusion, I think genomic capacity has greatly increased in Brazil over the last years, with dozens and dozens of laboratories, if not hundreds, generating sequencing data now. And although the infrastructure to generate pathogen genomic sequences can be extremely important for timely detection of emerging and re-emerging pathogens, there is, of course, room for improvement, and sustainable investments in research and development will be critical for this. There's also a need to better integrate genomic sequence data with COVID outbreak responders to guide and boost, for example, vaccination strategies. But this is not just for SARS-CoV-2. We could start going beyond that and look, for example, in using sequencing data in combination with other data sources to improve vaccination for influenza and for yellow fever. And finally, we really need to start thinking beyond COVID-19. What's coming next? We need more research at the animal-human interface to understand where and what are the zoonotic origins for disease X, and how we can learn. We need to start thinking how we can learn from the ongoing pandemic so we can actually be better prepared for the next one. I think I really want to thank Esther. Has been a pleasure to work with Esther throughout these last years, and some key PhD students that have done lots of the work presented here, especially Ingrid Claro and Darlan Candido, but also Jacqueline Goiz, postdoc in our team, and of course FAPESP and MRC for the key funding for this. Thank you very much.
Thank you very much, Nuno. That was as well a great talk, showing us some interesting aspects of the advancement of knowledge and its application, right? How knowledge in one thing leads to another, and then leads to another, and leads to people who can deal with something dangerous that appears suddenly. So you started from Zika to yellow fever, and then SARS-CoV, which is a world ranging problem. I'm sure we are going to have many questions and time for discussion in the end. Thank you very much. So I will now go on to our last speaker, who is Professor Fernando Sendes, medical doctor in science. He's a professor of neurology and coordinator of the epilepsy surgery program at Neuroimaging Lab at the University of Campinas in Brazil. He also directs a research center, which is the Brazilian Institute of Neuroscience and Neurotechnology, which is funded by FAPESP, the São Paulo Research Foundation, through a BOLD program that offers funding for up to 11 years, with several tough reviews that Fernando has faced during the duration of the center. And Professor Sendes is going to talk to us about cutting-edge research in neurological disorders. Please, Fernando.
Thank you very much. Thank you very much, Brito, for the introduction. I want to thank the embassy for organizing this event and for inviting me. I will share my slides. So in the few minutes we have, I will give you a brief overview of the work that has been done from the center that Professor Brito just mentioned, the BRAIN, which is a Brazilian research institute funded by FAPESP. We are in Campinas, state of São Paulo. And our goal with this project is to develop new methods and techniques to improve knowledge for diagnosing, treating, and preventing this debilitating disease affecting the brain. And we focus there are several conditions we work with, but we focus mainly on epilepsy and stroke, but also the studies go to other neurodegenerative diseases in the brain also. So we are a large group of researchers with many graduate and postdoctoral fellows. And the fields involve from medicine and biology, but also a lot of engineering, mathematics, computer science, as you will see. We have several partners in Brazil, but also a lot of international partners, including UK and other countries, as shown here. And today it's impossible to do a cutting-edge research in neuroimaging without working with big data. And for that, we need international collaboration, and that was what we are also doing. So our research, it goes from translational research. We have groups working on animal models to see the basics, concepts of the disease. That includes imaging, but also molecular biology and genetics, and also working with in vivo imaging of the brain and other modalities to understand the diseases. For example, epilepsy has several causes, and some of the patients, and in fact, many patients, do not respond well to medication, and they need to go for surgical treatment. We take the opportunity for this situation in that we have to investigate where the focus is in the brain and try to devise resection that can ameliorate or cure the patient. And for that, once we take a piece of the brain in this procedure, part goes for the diagnosis and part goes for research, and then we can investigate for example, some biological underpinnings in these conditions. This is one of the conditions. It's called focal cortical dysplasia. It's an embryological problem that occurs in the brain, and it's not very well understood. This is one study showing that earlier failure of the differentiation of these neuronal glia leads to abnormal neurons that fire and cause epilepsy. As I said, many patients do not respond well to medication. We need better tools to prevent or to diagnose early. And so we work on prediction models and algorithms that can, based on several features, try to see in advance which are the patients we will need, for example, surgery early on instead of the others who will respond well to the medication. With the support of FAPESP and together with other research centers in Brazil, we launched several years ago this Brazilian initiative for precision medicine, which is a genomic data sharing to look at underrepresented populations. And this is being a very well interesting source of data. For example, this is a study showing that the distribution of the local ancestry in our populations is different from the populations where we have international databases. And so when you're looking at complex genetic diseases, you cannot compare our population necessarily with data banks that are European or North American, for example, because there are differences in terms of the ancestry of this population. So those studies are very important. I said in the beginning that big data is crucial. Working in collaboration with big data has transformed fields such as physics and genomics, many others. And it came a little bit late to neuroscience, especially with the advance of neuroimaging, modern neuroimaging that can allow us to have tons of data to analyze. And so we use different levels of artificial intelligence models, including machine learning, deep learning, and many other aspects to try to dissect and analyze the features shown in neuroimaging. So by combining different kinds of data, we can help to understand the classifications, for example, patients automatically with epilepsy from healthy controls. We can detect lesions that need a very high expertise with the machine, and therefore this can be widespread to other centers that do not have this expertise. We can predict the seizure outcomes, as I will show a couple of examples, and this will help the better diagnosis. Also, we can understand better conditions in the brain. So this is an example of a very successful collaboration, which is the Enigma Consortium. In this case, this paper looked in a huge number of subjects, more than 51,000 individuals, and we tried to, this work tried to understand the relationship between the brain structures using MRIs and also with many other databases to look at the genomic locations of the areas in the brain that are developed according to this genetic features, and also predisposition to several common conditions that are affecting the central nervous system. Talking about epilepsy specifically, this consortium from Enigma has been able to bring very important new information with regards to the structure of the brain and how these structure abnormalities affect the condition that is very frequent in the population. So epilepsy affects at least 1% of the population. In underdeveloped countries, a little bit more than 1%. So it's a condition that it's very prevalent. So we can use the concept of artificial intelligence to, for example, try to teach the machine to get a very complex neuroimaging data, both functional and structural, and try to develop models that can, for example, classify subtypes of epilepsy for diagnosis purpose. And also we can go, and we are coming to this in a very good way in terms of being able to move from group analysis data to the individual analysis. So this paper showed that this algorithm can identify lesions that were not visible by naked eye in the examination. And these algorithms could identify focal cortical dysplasia, the lesion that I just talked about, which is a major step in terms of diagnosing those patients which usually have very refractory seizures and need operation to ameliorate them. But also we need to select the patients who will do well for surgery, because surgery does not succeed for everybody. So it's very nice if we could have reliable ways to do predictions. For example, who are the patients who will do better for surgery? And this is one example that automatic analysis using cortical measures from neuroimaging data can show a very good prediction for patients who are operated in the frontal lobe, but also in the temporal lobe, different forms of epilepsy. So these models are very important to advance further not only our understanding, but also the practical purpose for treating these patients. So in another area of our center, we are also trying to develop new techniques that could be applied to those conditions that I just talked about. Here I'm showing two examples, there are more. One is the development of a system which can measure the metabolism and the hemodynamics in the brain. But the good thing is this is not only non-invasive, but also it can be used in the bedside. So the patient does not need to be moved to another place to do a brain scan. So this can be as a monitor in the bedside. So this will change enormously the monitoring in ICUs for these patients with brain disorders, for example. And here's another example of advancement of a software that can put together different modalities of imaging, structural, metabolic, here glucose metabolism in the brain, and can show to the examiner in a 3D in very fast way where the lesions are. So we can, for example, plan the surgery and do a sort of a virtual resection and predict what is the surgeon is going to do in the OR. There is also, lastly, an area that we are concerned about rehabilitation and also improving mobility of patients affected by stroke and other degenerative conditions. This is an example of virtual and augmented reality that is being used for rehabilitation. And this is BCI using to command a wheelchair for patients with tetraplegia. And this is one of the success incubated companies that came from this group. And it works with virtual reality and also artificial intelligence for helping patients in terms of capturing behavior. And this can be used not only through assisted technologies for patients without mobility, but also for rehabilitation in several conditions. And this is already went to a commercial company, which has been very successful. So to move forward in this type of research, we need to integrate people, centers, and different areas of knowledge across countries, sharing data and sharing knowledge to advance further. So I want to thank the support by FAPESP, and thank you for the attention.
Thank you very much, Fernando. I'm having difficulty in turning on my image here, but thank you very much for a great thank you very much for a great talk. That was really, really impressive how you described this center, which goes from basic science to applied science to translational science to equipment to startups. It is really a great demonstration of the capabilities that exist in many regions of Brazil to perform research which not only is relevant for the patients, but is relevant for the economy, relevant for the country. Congratulations. That was really, really very interesting.
Thank you.
Now we are going to go to the Q&A session, and while we wait for well, there are questions in our Q&A board, but I will start by asking from Professor Sabino if she could tell us a little bit about the possibilities which obviously exist and existed, as you have shown, and the challenges of having this interaction between advanced research and the public health system. Because as you have shown, your research is advanced and it helps the public health system. Tell us a little bit about how does this happen and which are the challenges and the strengths?
Well, there are more challenges now. This interaction between the university and the public health system, because it's a little bit different than what I see in the US and in England, that the infrastructure is already set up. So there is a way that the scientists heard, and there is like an infrastructure of transmission of knowledge. And we are still behind this in Brazil. There is no, I see this a lot in the US, that they have already defined who are the scientists, how you choose the scientists who are going to talk to the FDA or whoever is going to take the decisions. And I think this is an important area for us to work, which is who is going to give the opinion. It can't be the scientist that the person in charge chooses. So you have to have a group, a way, and say, well, if you want to follow science, this is the group chosen, and you may change every 2-3 years, but that would be something really important. And I think different areas of the government should have this kind of interaction. And somehow I see it more clear in the US and the UK. So this is something that we should move to improve this capacity and understanding of what scientists are saying, how they are reaching their point of view.
Thank you very much, Esther. Now we have here in our Q&A board a question for Nuno. One of the attendants would want to hear from you about where we are at the connection between Zika and microcephaly.
I think, yeah, so that's a great question. I think lots of research has been put on hold on other viruses once we came into the COVID pandemic, which for us, for example, is something that we are really interested in using the last months from CADE to sort of come back to those research questions. Some of them involve questions related with Zika and microcephaly. But I think that so from the beginning on, there was a clear temporal and spatial link with Zika infections and microcephaly. And previous work in 2016 already has shown that there was a lag of around 14 to 17 weeks between the peak of Zika in a given place and the peak of microcephaly, suggesting this strong temporal association that most women that would deliver babies with microcephaly would be infected in the first trimester of their pregnancy. We did subsequent analysis showing that there was only a fraction. I can't recall the numbers exactly now, but there was a small fraction below 0.1% of those women that were pregnant and infected with Zika that would deliver babies with microcephaly. But there was also significant evidence showing that in lots of the places where Zika had passed, there was, like Esther has mentioned, the herd immunity threshold. The same is true for Zika virus, right? And the herd immunity threshold for Zika should be around 60 to 70% as well. So what we've seen in 2016, 2017 was this big epidemic throughout the entire country and most of Latin America and many other countries in Asia and a few in Africa. But we shouldn't be expecting big epidemics from Zika for the foreseeable years to come. So within, for example, I think the last estimates I heard was within the next 20 years, what will happen is that we will slowly increase the proportion of susceptible population that is going to be born, for example, that hasn't been exposed to the virus, and then generate somehow a sufficient number of people that haven't yet been exposed to the virus that is sufficient to generate again a big Zika epidemic, but that is still to come. I think we need to keep an eye. We need to keep an eye on that, but and come back. It is my opinion that I think we really need to come back to some of the more basic arbovirus research as well and see whether Zika has managed to persist in different pockets of the population. There's some evidence that Zika also became established in non-human primates potentially in Brazil and has been recently found in other mosquito species that is not linked to Aedes aegypti or albopictus. And we need to keep an eye on that because if that's true, then the consequences of having a spillback event from Zika in Latin America, as we've seen in yellow fever many centuries ago, could have some unpredictable results in terms of association with Zika and microcephaly for the future.
And let me see if I understood correctly one point in the central point in your answer. You mentioned, you argue that the Zika epidemics went away because people enough were infected.
Yeah.
That is the thing.
Yeah. Yeah. Brazil had a high number of reported cases, but like what Esther has shown, when we look at seroprevalence studies, lots of these locations where seroprevalence studies were conducted, which was just a tiny fraction of municipalities where this happened, has shown that over 60, 70% of the population was exposed after 2016, 2017 epidemic there.
But does that mean that the disease didn't spread geographically in Brazil? Because the high incidence was in some cities, some regions of Brazil.
The spread of Zika is contingent on the abundance of Aedes aegypti, and it's dependent on temperature and humidity, which drives the abundance of these mosquitoes, right? These mosquitoes live in cities, so you wouldn't expect to see many cases outside of cities as well, or as many cases. And there is a clear seasonal pattern as well for Zika, but it spread very much across all the 5 regions, maybe less in large urban centers or for example, in the São Paulo city. I had fewer cases. There were some cases that were detected, but there's still fewer cases. And that's something we don't exactly understand why. The same for chikungunya, which is another mosquito-borne pathogen virus that spread very rapidly across the entire country through an introduction, most likely from Angola in 2014 in the Feira de Santana, and then the virus spread very rapidly throughout Bahia, Northeast Brazil, and so on and so forth. But we still don't know why chikungunya hasn't caused a big outbreak in São Paulo, and that's one of the questions that we are also interested in looking at, maybe combining surveillance in mosquito populations, which is challenging, of course, and more time consuming, and also combining that information with, for example, information like from the REDS cohorts that Esther is leading. That would be really interesting to understand changes in temporal patterns in disease incidence for this arboviral disease over time. And I think once we combine these different sources of information, we'll have a better idea why it spread faster in the northeast region of Brazil, because probably there's also a higher mosquito suitability in these areas, leading to higher abundance of mosquitoes. But certainly other factors are also possible, and other factors could also help to explain why it hasn't spread so rapidly or in other parts of the country. And it has been proposed that there might be some interactions with other previous exposure to other mosquito-borne viruses as well, especially for chikungunya. And there's also some suggestions that there is an interaction with dengue, previous exposure to dengue. So we're still trying to figure out if there's some protection conferred by recent exposure to dengue for Zika, and for chikungunya, if there's any other relations with any other viruses like Mayaro. But definitely those are very interesting questions and worth pursuing in the future.
Thank you very much, Nuno. I would have now a question for Fernando, which is, in one of your slides, you showed about the part of the project which relates to genetic diseases and to the study of the Brazilian population genomics. And could you comment a little bit more about the relevance of having this kind of study, considering the differences in the genomic characteristics of populations across the globe?
Yeah. For example, when you're dealing with the complex diseases that may have very rare mutations, a mutation can be something beneficial or normal. A mutation can be pathogenic. Okay, so the definition of a pathogenic, it's you can use a statistical definition. It's something that is not seen in the population, in the majority of the population statistically. So you can go further and look at it. Once you detect it, then you're going to do another kind of studies to do that. Well, a given normal variant that we say in a population can be the prevalence of normal variants. They vary according to the population, because it depends on the ancestry. So in Africa is one thing, in North America, in the indigenous population, and so on. When you take a population like Brazil, that is very much admixed, and it's very different because of that. So our proportion of mixture with African descendants, the indigenous populations in Brazil are different from the North American, and so on and so forth. So this makes this assumption to look for very rare conditions very difficult if you don't have a database to compare with. And so far, we did not have a database. The big database comes from Europe, from North America. So we are looking for things that are in our population, and you don't have a comparison. Mm-hmm. So we need to have the data banks from our population to look for this. Otherwise, some of the search for diseases will be meaningless. I think the same thing applies for example, other diseases. We heard a lot about virus, and also the susceptibility to be infected is also dependent, as in any disease, to the genetic background. So perhaps this can also explain some of the differences in the populations from different countries and places. So it's a very complex issue.
Thank you very much. Yeah. And let me follow up with another question that came here for you, related to the breadth of the research in the center that you lead. Do you, in that going from basic to applied to translational to startups to equipment, software, everything, do you, how large is the team in your center, and do you have foreign researchers who came to work there, who you brought in with local support?
Yeah, we have a huge collaboration, not only inside the university, but also with other universities, as I showed very briefly. But also we have very tight collaborations with international partners. And we have had people visiting us, staying sometimes. So many of the projects are in collaboration, very close collaboration. So we have many students going abroad, and others, we're receiving students from UK, from other places. So there is a very, very good interaction between these kinds of research. So yes, we had people from many places, some of the places I mentioned briefly, that are coming routinely. And also there are some FAPESP initiatives that you know very well, of course, so the sprints and other that we've been using a lot with Australia, with many other places. And that is being instrumental, because the share of knowledge from all the sites are fundamental for this kind of research.
Thank you very much. Nuno, I have here another question for you, which is about your views and impression about international collaboration in Brazil. You could give us a view of a person from Europe, from the UK, that has done extensive interactions and collaborations in Brazil. I think you mentioned that you traveled, what was that, 2,000 kilometers in doing that mobile, yeah, 2,000, the Zebra, 2,000 kilometer mobile lab. Let us know about your views on this. You found researchers in many places, you found collaborators. How's that?
I can talk about my experience, right?
Sure.
But I actually spent 1 year at USP in Ribeirão Preto for my graduate studies, and then when I started doing my PhD, we were invited. We are part of a bioinformatics team that organizes VIMI workshops. It's called the Virus Evolution in Bioinformatics. Is the oldest workshops on virus evolution in bioinformatics. With I think it's going on the 25th edition this year. So it was started by my co-supervisor Annemie van Dam many years ago. So I've been teaching in these VIMI workshops since the beginning of my PhD. And in one of these events, we were invited by Wander Chagas to go to Ananindeua, so to IEC, to Instituto Wander Chagas, right? And essentially, they've invited the whole team, so we went there. And the next year, the funding, there was a cut in the funding, and they could only invite one person, and then they invited me. So we started a collab, well, because I guess the language as well. And I was always very interested as well by then in genetic analysis of viral pandemics. So my work was mostly done around HIV origins as well during my PhD, and how we can use these models to answer some key questions that are not just related to HIV, but to any virus really. So then I started. So I started as a visiting professor at Ivandro Chagas, and would organize together with Professor Marcio there, and we would organize sort of an annual workshop on virus evolution bioinformatics, mostly focused on arboviruses. And we started collaborating on dengue, first dengue 4, and then dengue 1, 2, and 3. And then chikungunya came in, and then Zika came in, and there was an exchange. So I would receive students. Then I moved to Oxford. I would receive students in Oxford. We trained them for a few months in Oxford, and we would have, like, I think, biannual trips to Brazil. And then I became a visiting professor at Fiocruz Bahia as well, and then at UFMG as well, and then now at the University of São Paulo with Esther. So it's been a long journey, but a very interesting one. So wonderful people, wonderful collaborations as well, and an absolute pleasure really to be there. With the pandemic, I think it became a bit more difficult because of travel restrictions, of course, but we were receiving students and are still receiving students. So currently, I'm hosting Jacqueline Goisch as well from Esther's team, and we have...
She was here in Oxford yesterday, right, in an event, yeah.
There you go, yes. I couldn't come, unfortunately, but so Jacqueline was there, yeah, and is here, and so my team is very much also very focused on, I mean, there's a big Brazilian community in my team here as well, and there's an interchange with Esther as well, and with other other co-PIs that we have also from the project in UFMG, for example. So we have an interchange of students, and we're using some of the international networks and funding opportunities to facilitate that.
Great, thank you very much. So you really know a lot about Brazil. So you started in Ribeirão Preto, where they have very good draft beer downtown.
I lived very close to Pinguim.
Pinguim, yeah, right. Everybody goes there. Excellent. Now let me ask Esther a similar question, but seen from Brazil. What were the impacts of the pandemic in the international collaborations that the center has in Brazil? Because as you described it, a lot of your work relies on international collaboration. And with the pandemic and the difficulties in travel, did you see especially hard to overcome difficulties, or did Zoom compensate a little bit for that?
No, I think it didn't, well, always a problem, but it is like talking through Zoom, or in the end, we could really organize now meetings that we kept more common, like routine. So we do have this week call with the CADE team that now includes many other people that are not in the beginning of the program. I think one of the difference with this project was that during the Zika was easier for Brazil to get money from outside, and because this epidemic is everywhere now, there are some constraints with resources for international collaboration, which also due to the political way, like in the REDS program during the Trump, it wouldn't give any money for any study outside. So we were very lucky to keep the REDS program with this idea. And I think this is important, that we should try to say how diplomacy is important in this area. We should try to support these teams that are working together in an epidemic. And this is the basic for the next epidemic. So I'm an example. I started with HIV. I was trained through a Fulbright grant. And this made, at that time, I think 2,000 people were trained with the same program. Let's organize the people on AIDS. So this moves across years, and we can then work with many other epidemics. So this is very important. And it has a lot to do with the science and diplomacy, how you do these groups and how you keep them working. And this is very important in all sides. And I hope in UK we will understand that and we can keep the Kali project together. So I think this is the message. How important is these moments, and we should try to keep the groups moving together.
That is really interesting. And you mentioned something that makes me think about the relevance of capacity building, of training young people to deal with problems in the future. Because I believe, you just said that, I believe Nuno mentioned that too, that he started his work in infection and viruses also during the HIV times. And also Dr. Fauci, right? Dr. Fauci was a big leader there in the fight against HIV. And so it makes me think that this pandemic is probably training a big generation of virologists for whatever is going to happen to us 30 years from now. And let's hope they are learning well, and that they will be able to take care of that, as well as scientists like you guys are doing now. Yeah. I have here another question to Fernando, which again relates to the breadth of activities, which is, it's a question about connecting artificial intelligence, machine learning, and what you are doing. And the question mentions that it must be challenging to have the computer scientists, the computer engineers, and the medical doctors and the biologists talking the same language. How does that happen?
Yeah, that's a very important question, and that makes a lot of difference in terms of doing something meaningful and not in this field from now on. This is because it's very difficult to talk across disciplines, but once you get the know-how, and once you are open to listen to different fields of knowledge, then actually the research work gets much more interesting. Because every day I learn more with my colleagues who are mathematicians and physicists and computer scientists, and I'm sure they learn a lot with the neurologist, with the physical therapist, and so on. So we have in the lab, it's a very dynamic lab, because you have people from all sorts of backgrounds, and with time they learn how to, interact. And I think my view on that is the rule is the following: you don't need to be an expert of artificial intelligence programming, for example, but you need to know the basics, a little bit of the basics, to have enough to talk meaningfully with your other collaboration, and vice versa. So everybody here who are physicists and engineering, they know quite a lot about the brain, the function of the brain. So they learn all the way. I have several examples, in other places that, for example, in the place where I trained in Montreal, one of the professors there, he was an engineer, and he's one of the persons that knows more about epilepsy than most of the neurologists that I know of. And that's why he developed very meaningful research. He was pioneer, for example, on the EEG to build up programs that can detect seizures automatically. So this changed the whole field about the diagnosis and so on. So what you need is somebody that goes and learn a little bit more about the other science and then integrate this. So it's very fun. If those who are listening did not try that, I would suggest to give a try, because it is very, very good.
Thank you very much, Fernando. Now we have 1 minute for a last question that came for Nuno, which relates to one of your slides in which you had a graph showing what seemed to be two pandemics, one on top of the other. There was a blue curve and then there was a pink curve. Was, I think, the posterior density of virus lineage movements. Is that really, can we think of that as two pandemics with a different, as if they would be different viruses or not?
I mean,
you are on there.
No, so yeah, that's a good question. No, so this is essentially two phases or two periods of
okay.
that essentially look at the establishment of one pandemic in a given country. So in this case, of course, in Brazil. So the first phase in blue shows the timing of the different hundreds of introductions that we've detected from many other countries into all the regions in Brazil. And then second curve, which is in red or salmon color, essentially shows the actual establishment of the virus. So the virus first goes from a phase where you have multiple introductions in a given country, right? Let's say we had multiple introductions from Italy to São Paulo, and then a person who is infected in São Paulo might or not transmit that virus to another person. On average, it transmits to, especially before the implementation of non-pharmaceutical interventions, it would be spreading, like Esther mentioned, with the R naught around 3 or 4, it would be spreading to 3 or 4 other people. So what happens is that in the initial phase of the pandemic, you have mostly international imports that may or not lead to local transmission. And then in a second period, which is the other salmon curve or red curve, if you like, posterior density, we looked at the essentially the timing of virus lineage movements within Brazilian locations. So what you have there is just migration of the virus within the country. So no longer needing the seeds, the international seeds, but essentially sort of suggesting already a well-established local epidemic with community transmission in the country. So it is as if like you have these seeds that are required to establish the first phase of the pandemic, and then those seeds sort of flourish, and then you have multiple transmission chains, but they're only circulating locally, and it covers those two phases. What I think is interesting is that, I mean, so we know that lots of these analyses are bound by biases in specifically biases for heavily sampled data sets that target or a specific group or a specific location. But what we've tried to do here was slightly different, a different thing. So we managed to look at the number of cases from each of the states in Brazil, and we've managed to select randomly a proportional number of sequences to the number of confirmed cases and generated a representative data set. And with that regard, so we were able to, and because we've done that, and we've used the representative data set, we were able to estimate these parameters a bit more accurately than in other studies. And I think this was really the first examples of a representative data set, setting up like the bar high for other studies that were done inclusively in the UK. So that were done afterwards.
Thank you very much, Nuno. Well, thank you all three very much. That was an exciting, informative, and interesting session about modern science or up-to-date science. And I will now give the floor back to our organizers at the embassy for a closing session of the symposium.